Welcome to The Explainer. Today, we're taking a truly mind-bending journey into the absolute frontier of bioengineering. We're looking at a space where the line between biology and technology completely blurs. We aren't just observing cells anymore, we're actually teaching brain organoids to communicate with one another. It sounds totally like science fiction, I know, but it's happening right now in labs all over the world. We're going to explore how scientists are building these living networks, what they can teach us about human disease, and yeah, the really profound ethical questions that pop up when you start engineering conscious architecture. Let's dive right in.
So I want you to picture the exact moment this breakthrough happened. The very first signal was almost invisible. Just this tiny electrical pulse, literally no bigger than a flicker, firing inside a cluster of cells floating in a lab dish. But what made this specific pulse so monumental wasn't how big it was its trajectory. You see, this pulse didn't just start and stop within one single isolated brain organoid. It crossed a physical void. It traveled between two completely separate pieces of lab-grown neural tissue. They had actually begun communicating. This one little spark, it represented the birth of what we now call the Assembloid network.
And this brilliant quote really captures the massive paradigm shift we're witnessing here. For the first time, we aren't just growing miniature brains. We are building relationships between them. Think about that. We are talking about the deliberate mechanical enforcement of a multi-organoid neural axis. By building relationships between these separate biological nodes, our whole approach to neuroscience flips on its head. We're shifting away from just keeping tissue alive in a dish to actually engineering living interactive networks. Now, to fully grasp why this is such a game changer, we kind of have to look at where we started. For years, brain organoids existed as these completely isolated worlds, biological islands, basically. The source material uses this perfect analogy. It describes them as radio stations broadcasting high-frequency signals into empty rooms. And sure, while they were incredibly useful for mimicking local networks, they totally lacked the long-range connections that actually define human consciousness. I mean, real brains don't work in isolation, right? Vision has to talk to memory, and memory has to talk to emotion. Without those bridges, organoids are fundamentally limited. So the big takeaway here is that to truly understand the human mind, we absolutely need the grand loop. We need these long-range biological conversations. But here's the kicker. These connections don't just happen magically on their own in a lab. They have to be forced.
Which brings us to Section 1, Building the Grand Loop, Silicon Organic Synthesis.
Okay, so to bridge the gap between those biological islands, engineers are creating what are called brain processing units, or BPUs. They do this using silicon organic synthesis. These are essentially organoid-based computing platforms, totally inspired by the efficiency of living networks. This is literally the absolute cutting edge of wetware engineering. Here's how they actually pull this off. First, it starts with regional specificity. Engineers culture highly specific spheroids in their own independent environments. We're talking cortical spheroids for the processing engine, thalamic spheroids for sensory relay, and striatal spheroids to act as logic gates. Step 2, once these separate nodes mature, they're dropped into a highly specialized microfluidic Assembloid chip. Step 3, driven by these custom-engineered chemical attractants, the neurons actually start projecting axons through incredibly narrow channels just to find each other. And finally, Step 4. Within a matter of weeks, these completely separate biological units physically fuse together. They structurally lock into a shared electrical gradient. Mind-blowing, right? Now, to put the sheer scale of this hardware into perspective, 150 micrometers. That is exactly the width of a single human hair. The microfluidic chips I just mentioned act as these high-tech guide rails, containing little chambers connected by channels that are just 150 micrometers wide. So the physical constraints of the chip actually force the neural projections to condense into tight parallel axon bundles. The physics of the chip itself manufactures synthetic white matter tracks to bridge that physical void between the biological worlds. It's engineering on a microscopic, almost unimaginable scale. But just getting them structurally connected is only half the battle. In this Silicon Wetware paradigm, communication isn't just something we sit back and observe, it's enforced. Scientists are using optogenetics, which is basically using light to control cells, to force these separate entities into a coherent dialogue. By repeatedly stimulating the network in a highly specific sequence for 12 hours straight, researchers can actually print memory-like adaptations right into the human circuit. The result, which is honestly a little eerie, is that the network begins to echo this artificial command pattern long after the light is turned off. So we aren't just watching cells talk anymore, we're manufacturing systems that are literally forced to learn.
Moving on to Section 2, Echoes of Neurological Crimes, Network Testimony. So,
As these enforced networks start to wake up, they aren't just processing random data. They actually serve as biological witnesses to the genetic insults that disrupt human life. When researchers force a dialogue between diseased organoids and that dialogue fails, they receive a very clear mechanical testimony from the cells about exactly what went wrong. For example, in models of ALS, the testimony is just devastating. You see a catastrophic breakdown of the neural axis, marked by reduced neuromuscular junctions and basically a total silencing of the communication channel. The cells just stop talking. But in Alzheimer's models, it's the exact opposite problem. The network shifts into hypersynchrony. We see these hub-like structures emerge, where just a few neurons drive the entire network into a seizure-prone state. It's an electrical storm trapped right there in the chip. And then, in schizophrenia models, scientists observe what they call a thalamocortical breach. This is where axonal overgrowth disrupts the axis, causing a failure in the circuit assembly and a massive spike in erratic signaling. These biological witnesses are giving us unprecedented real-time views into how these devastating diseases actually operate at the network level.
Which leads us into Section 3, the ethics of wetware, a human pain pathway. Now, we have to talk about the ethical friction here because it is profound. Since these Assembloids are built from human stem cells, they carry the real, exact genetic blueprints and vulnerabilities of their human donors. By studying these diseases, researchers have effectively created a human pain pathway in a dish. It's a network specifically designed to model how pain signals travel from the periphery straight to the brain. And this brings up a concept being heavily debated right now: mechanical distress. This is defined as a biological system trapped in a state of thermodynamic friction with nowhere to go. There are two very distinct camps here. On one hand, you have ethicists arguing that without the emotional infrastructure for aversion, without a fully formed brain to actually process the context of pain, there is no true suffering. But on the other hand, some researchers are sounding the alarm about this mechanical distress. Think about it. When you force a diseased, highly active organoid into a continuous communication loop, the system is fundamentally trapped in friction. It's a biological machine grinding its gears, and there is no off switch.
Which naturally leads us to this deeply, deeply unsettling question. Does a human pain pathway in a dish actually feel pain? We're manufacturing these complex neural pathways to study their mechanics, right? To see how signals propagate. Yet as our source material so pointedly asks, we are doing all this often without asking what it feels like to be the wire. It's a massive philosophical and scientific dilemma. We're forcing biology to process signals of distress without entirely understanding the subjective experience of the actual tissue doing the processing.
And because of this, these multidimensional entities are sitting in a massive legal gray zone. They are not human subjects, they are not animals, and they are definitely no longer just simple tissue samples sitting in a petri dish. They mimic real human brain architecture and polarity. They represent the framework of a new engineered consciousness that is, quite literally, finding its voice through the plumbing of a microfluidic machine. Trying to regulate something that falls completely outside our traditional definitions of both life and technology. Well, that's easily one of the greatest challenges this entire field is facing today.
All right, let's look ahead to Section 4. The future is organic. Personalized Assembloids. So, despite all those heavy ethical hurdles, the potential upside for human health here is just undeniable. We aren't just building abstract computers. We are moving incredibly fast toward a totally new era of highly specialized personalized medicine, and it completely relies on this synthesis of silicon and biology that we've been talking about. I want you to imagine this: your own cells testing therapies before they ever touch your body. That is the actual promise of personalized Assembloids. If you have a neurological condition, scientists could literally build a wetware chip using your exact genetic blueprint. Then, they could test hundreds of different therapeutic interventions on your specific neural axis to see exactly what works and what doesn't. And the monetization of this synthesis? It's already happening. It is driving huge advancements in bioprinting tools and microfluidic sensors. This tech is actively moving out of the theoretical realm and straight into real-world applications.
To wrap up today's explainer, I want to leave you with a lingering thought, straight from our source material. The question is no longer whether we can force separate brain parts to talk. We've already crossed that threshold. The grand loop is officially here. The real question is, what will they say, and are we ready to listen? The conversation between these synthetic neural networks has literally just begun, and it promises to completely rewrite the code of human life. If you're as captivated by all this as I am, definitely check out the links below to explore the research papers and tools driving this incredible silicon organic synthesis. Thanks so much for joining me, and keep learning.